Okay, so in this video, I'm going to cover homeostasis, negative feedback, and blood sugar control. So homeostasis is the idea that you maintain the internal conditions. Now, you should have come across this at GCSE, so it shouldn't be a new concept. What we're going to look at is how blood sugar control is maintained, and in a different video, we look at the level of water in the blood, or the water potential of the blood. So we're talking about things like blood glucose, which, as I said, we will cover. We're talking about the water potential of the blood, or the concentration of the blood. You could be talking about pH. You can even be talking about blood pressure. The only ones we're going to talk about specifically are the first two, and the one that we're going to do in this video is this particular one here. Now, homeostasis can be controlled by both nervous system and the hormone system. And in fact, obviously, when we're looking at water potential of the blood, we're talking about hormones. And when we're talking about blood sugar, we're also mostly talking about hormones. But if you were to look at temperature, which is another example of homeostasis, although it's not on the specification, you would be talking more about the nervous system, which will be covered in a later video. So we need to think about the process of negative feedback. Now, negative feedback is the way in which our internal conditions are controlled. So what does negative feedback actually mean? Well, in terms of what it means, it says that if the internal condition deviates from the normal, so in other words, it goes away from the normal, the body responds and brings it back to normal. So, for example, in the case of body temperature, if your body temperature goes too high, above 37 degrees, the body will respond, for example, with sweating or dilation of blood vessels, and bring the temperature back to 37. And that's what we mean by the process of negative feedback. And we're going to illustrate it here with reference to blood sugar control. Okay, so let's think specifically here about the control of blood sugar. Now, you will remember from GCSE that whenever you're talking about homeostasis, you're always going to be talking about the idea of having a receptor, something that receives the information, something that coordinates the information, something that responds to the information. Now, control of blood sugar is a hormonal control, and it involves two hormones that you've come across before, insulin and glucagon. So both of them are secreted by the pancreas, but more specifically, insulin is secreted by the beta cells of the islets of Langerhans in the pancreas. So within the pancreas, there are these groups of cells called the islands of Langerhans. And within those islets, there are alpha and beta cells. Insulin is secreted from the beta cells, whereas glucagon is secreted from the alpha cells of the islets of Langerhans in the pancreas. Now, just to summarize what we already know from GCSE, we know that those hormones are responsible for the conversion of glucose to glycogen, which is the polysaccharide that's stored in the liver. So we learned in the molecules unit that you get alpha glucose. Alpha glucose is joined by glycosidic bonds to create glycogen, which is stored in the liver. So it's a polymer of alpha glucose. Now, that is a process that can go in both directions. So if we think about insulin and glucagon, insulin causes the conversion of glucose to glycogen, and glucagon causes the conversion back again. So let's think about what is happening in terms of when we produce it. So we're going to do this as a flow chart. So we're going to start with blood sugar or blood glucose increases. The blood glucose level goes up above what is considered as normal. That will be detected by the pancreas, because the pancreas is the receptor for blood glucose level, and that will secrete insulin. And we know that insulin is coming from the beta cells of the islets of Langerhans. Now, the insulin will travel in the bloodstream, because that's how hormones travel, and will go to the target organ, which will be the liver or the muscle cells. Okay, these things happen in both cases. And what does it do when it gets there? Well, the first thing that it does is obviously it binds to receptors. So it's going to bind to receptors on the liver and muscle cell. Now, remember that when we learned about proteins in membranes right at the beginning of the AS course, we learned that there were proteins within the phospholipid bilayer of a membrane that can act as receptors, and they are complementary to their particular hormone. So we're talking about a receptor on the liver and muscle cells that is complementary to insulin. When it binds, the first important thing that it does is it encourages or increases the number of glucose channels in the membrane. Okay, so in the plasma membrane, because that's the outside membrane of the cell. In other words, it increases glucose permeability. So the cells are now more permeable to glucose. As a result, more glucose is taken up by the cell. And there are going to be different things that happen as a result of this. The first thing that happens is we get an increased rate of respiration. So the cells actually respire more. And the second thing that happens is that we get more glucose converted to glycogen. Now this has a name. If you cannot remember the name, you can use the description that I have used before, but the name is glycogenesis, because we are creating, like Genesis, we are creating glycogen. Those two things, the increased rate of respiration so that you're using up more glucose, plus the fact that you're converting it back into glycogen, will mean that blood glucose level returns to normal. Now, what we should actually see is what often happens is it slightly overshoots the normal, and we then have to bring it back to normal using glucagon. But let's imagine it returns to normal, and that's the idea of the principle of negative feedback. Now, what about glucagon? Well, by the same token, let's imagine your blood glucose level falls. So this time, you've not eaten for a while, you've fasted, your blood glucose level falls. Again, that will be detected by the pancreas, which will secrete glucagon, all right, which we used to remember by glucose has gone, the glucagon, this time from the alpha cells of the islets of Langerhans. Again, it's a hormone, so it will travel in the bloodstream to the target organs, which are the liver and muscle cells, and again, it will bind to receptors which are complementary and specific to glucagon on the liver and muscle cells. And what does it do when it gets there? Well, it actually enacts what we call a secondary messenger system, but we will come to that in a different video. But as a result of the secondary messenger system, different things will now happen. So the first major thing that will happen is that the glycogen is now hydrolyzed to glucose. So we're breaking our glycogen down. That is called glycogenolysis. Lysis means to break, and you're breaking your glycogen. The other possibility is that things like amino acids or fatty acids are converted to glucose. And that term is called gluconeogenesis. You are making glucose from something else, gluconeogenesis. You will also, of course, get a decrease in respiration rate, although it's going to be the first two that are going to be the most important. Again, as I said, if you cannot remember the names of the terms, gluconeogenesis and glycogenolysis, doesn't matter, you can describe them in words instead, as long as you know one or the other. That will lead to blood glucose level rising back to normal. So if you were to look at a graph, for example, of time against the blood sugar concentration, okay, let's imagine that this was the normal level. What you would find is the person's blood glucose level is at normal, then they eat, their blood glucose level rises, then they'll start to secrete insulin that will drop their blood glucose level. It normally slightly overshoots, as I said, so then they'll secrete glucagon, which brings their level back up. They may overshoot again slightly, and so on, until eventually it settles back to normal again. Now, if a person is diabetic, as we shall see, their blood glucose level not only starts off higher, but also has trouble coming back down again, okay? And that's what happens in diabetes, because the only way it's going to come back down is with its use in respiration rather than its ability to be converted to glycogen. So that leads us on to thinking about the process of diabetes. So there are two types of diabetes. There's type 1 and type 2. Now, type 1, which is often called juvenile diabetes, because it tends to appear in childhood, either because it has started right early in childhood due to a genetic condition, or it appears slightly later. Whereas type 2 diabetes is late onset diabetes, it tends to appear in middle age and is linked often to obesity, but not solely linked to obesity. So what is the difference between type 1 and type 2 diabetes? Because after all, in both of them, the blood sugar level will rise, and it won't be able to be brought down normally. So type 1 diabetes means that they don't make insulin. So for some reason, the person is unable to make insulin. There's a genetic mutation in the insulin gene, for example, and therefore they cannot make insulin at all. So therefore they cannot convert glucose to glycogen. Which means that their blood sugar level would remain too high. That in itself is dangerous. It makes the blood really thick. It can't flow properly. And the tiny little capillaries in places like the eyes and the fingers, don't get the actual blood flow they need. And that can become a major issue. So how do we treat it? Well, it's very easily treated in terms of we can control it. It's treated with insulin injections. A person with diabetes type 1 will need to have injections of insulin regularly, 2 or 3 times a day, depending on what type of insulin. Now often done automatically with a pump, or it could be done through actual injections. Type 2 diabetes is slightly different. Type 2 diabetes, they make insulin, but the receptors on the liver cells don't respond. In other words, there's something not quite right with the receptors. It's not being able to bind to the insulin. As I said, it's often linked to obesity, to the liver being a little bit fatty, but it has the same effect. They still cannot convert glucose to glycogen. So the principle of what it will do is going to be exactly the same, but obviously you're going to have to treat it differently because it's no good giving them injections of insulin. They already have plenty of insulin. So it's treated by reducing the carbohydrates in their diet, so they don't actually take in so much sugar, and also by increasing exercise, because exercise will help to increase the process of respiration, which is their way of bringing the glucose level back down to normal. Okay, so in an exam question, they'll often show you a graph, and you have to say which one's got diabetes, which one hasn't, and or they might give you some descriptions. Is it type 1 or type 2? Sometimes they give you information about a blood glucose test that they may do and ask you to compare them.